Okay, welcome. This week we are going to do some experiments on how to kill bacteria. Now there's multiple ways to do that, but we're going to pick a couple different ways, and we're going to use some antibiotics and we're going to use some bacteriophage. And so there are a couple assays that will use the antibiotics, this diffusion assay, which is very simple, very cool. We will do minimum inhibitory concentrations also. We'll determine that for different antibiotics. And we'll look at the sort of host rate. All right, so antibiotics can be used to treat infections. They were discovered quite some time ago by Alexander Fleming. He had a nice messy bench over here, and what he saw on his bench was a plate that looked something like this, where he had bacterial growth, and then he had this mold growing, and then you could see the region where bacteria were not growing. So he took that as an indication maybe there's something coming out of this mold that's inhibiting growth of the bacteria. And they called it mold juice, which is not very good for marketing, but this ended up being penicillin secreted by this mold, and this revolutionized medicine. It made what otherwise would have been fatal bacterial infections now sort of routine. People will go take an antibiotic and survive quite well. So antibiotics have different mechanisms of action. There are some that we'll work with this week. We're going to work with ampicillin. Ampicillin is a beta-lactam, and it inhibits cell wall synthesis. And there are other antibiotics listed here that work in this manner. So here we have our cell, and then arrows pointing to the cell wall or to the cell membrane, to the ribosome, to things that happen in DNA, etc. We're also going to use kanamycin. It's called an aminoglycoside. It binds to 16S ribosomal RNA in the 30S ribosome subunit. It causes misreading of mRNAs, so you get non-functional peptides, and this results in killing of the cell. A couple other that we're going to use are erythromycin, it's a macrolide. It binds also to the ribosome, but to 23S ribosome RNA, which is in the 50S subunit of the ribosome, right here. This inhibits peptide chain elongation, so you can imagine how that might be bad for a cell. It blocks the peptide exit from the ribosome. Chloramphenicol binds also to the 23S ribosomal RNA in the 50S ribosomal subunit, and it again inhibits peptide chain elongation, and this inhibits peptidyltransferase. So some things about antibiotics. So over the past few decades, we've seen a decline in new antibiotics that are being discovered. Now this is coupled with a rise in antibiotic resistance roughly over that same time period. So this is now becoming an impending catastrophic event for humanity if all of a sudden antibiotics will not work as well as they have because bacteria are becoming resistant to them. And if we're not discovering new antibiotics, it's going to make it even harder to treat infections. Resistance, the thing about antibiotic resistance is it develops pretty quickly. So here is just sort of a timeline. From the top is older to more current times on the bottom. And so in blue is where the darker blue is where antibiotics have been introduced, and so here's penicillin. And then in the darker blue regions, penicillin is used fine, and then all of a sudden, when we reach this lighter blue area, that's where antibiotic resistance was first observed and then has continued until present day. And so you can see that for all of them, there is different times where the antibiotic was introduced and used fairly well, and then resistance arose after that. For methicillin, it was a very short amount of time, and for the other ones, you can see that they have different times where they were effective. And most of them are still effective. The antibiotic resistance is not ubiquitous, but it is getting worse. So how does antibiotic resistance occur? So we'll talk about it on different levels. So if we have a whole bunch of bacteria, a few of them are going to have naturally sort of resistance to antibiotics that have come about through some sort of mutation. We'll talk about different kinds of mutations that can happen. When we use antibiotics, the antibiotics will kill most of the bacteria. And so we see these fainter gray ones, these are the bacteria that have been killed. But those that are resistant, these orange ones, will survive. Now, when this happens, the population dynamic has changed. There used to be, in circle 1 over here, there was mostly sensitive bacteria shown in blue, and only a couple, or very few, resistant bacteria. When those sensitive bacteria are gone, now the resistant bacteria will repopulate whatever environment that they're in, and most of what is going to exist after these bacteria start to regrow and repopulate will be bacteria that are now resistant. And so this happens through a mutational pathway, but there are also some pathways where multiple resistances can be transmitted from one bacterium to another. Okay, so how do bacteria, what mechanisms do they use to resist antibiotics, to sort of survive in the presence of them? And so what we have here is there are mutations are going to occur that alter the bacterial antibiotic interaction. And they can be thought of generally in 5 different ways. The first one that we're going to talk about is decreased uptake. So simply, the bacteria does not let the antibiotic in. It changes either its membrane or receptors on the surface of the cell, and the antibiotic does not get into the cell. And the antibiotics are shown here by these little sort of red and white pills. Other ways that it can become resistant is if it does get in, they have pumps, efflux pumps, which can then basically pump it out of the cell. So they just get rid of it. It enters the cell, and then they can get rid of it. And the specificity of these pumps can change by mutation. The way we mostly think about this happening is that antibiotics there are enzymes that will break them, that will inactivate them in some sort of way. There's an enzymatic reaction that the antibiotic no longer is functional. It gets cleaved into parts or in some way degraded. Slightly more complicated way that things can happen is normally an antibiotic may work through a certain pathway. Now, if that pathway, if that antibiotic now gets degraded by that pathway, it can find another pathway and go through an alternative enzyme, an enzymatic pathway. And then some of the ways that happens is that simply there are the target sites inside the cell, be it the ribosome or other molecules that the bacteria gains mutations that change in the position where the antibiotic would interact with them and inactivate those macromolecules. Okay, so those are many different ways that this can happen, and the genes that do that can either occur by spontaneous mutations that happen in the genome of those organisms, but that's kind of a slow process. What's been happening is that bacteria can transfer plasmids that contain multiple genes that confer resistance to different antibiotics. So you can have a genetic transfer of material between bacteria. And it doesn't have to be antibiotic resistance genes. It can be many different kinds of genes, but those are things, when they are transferred, it increases the frequency of those kinds of genes in a population. And the transfer doesn't have to be between identical species. It can be transferred between different species. And this is something that is of concerns, and CDC has put this out just last year in 2019. And so these mechanisms that I've talked about in genetics and evolution class that many of you may have heard about are still a concern. So transduction, where bacteriophage mediate the transfer of genetic material between bacteria. Conjugation, where bacteria contact each other through a pilus, or through pili, and transfer plasmids between bacteria. And then transformation, where bacteria die all the time, and when they die, they eventually break open, their DNA gets released and just laying around in the environment. And some bacteria, they like to take that DNA and bring it into their cell and incorporate it into their own genome. And when that happens, if those happen to be antibiotic resistance genes in that DNA, then the bacterium, the recipient bacterium, this guy up here, can take those genes and then use them to its own benefit. Okay, so how are we going to look at bacterial interactions with antibiotics? So one thing we're going to do is a very simple, just nice, clean and easy assay called the disc diffusion assay, also called the Kirby-Bauer assay. And what it entails is you take an agar plate and you spread bacteria all over it. One bacteria that you want, so you want to work from a pure culture of bacteria, and we will provide you with those. So you spread the bacteria all over the plate, and then you take these little discs. Now the discs can have nothing on them, and we'll have controls of discs that are just little paper discs that we'll put on there. But then we also have discs that contain antibiotics at very specific concentrations. And the idea is that you put those discs onto, if they have antibiotics, you put them onto the plate, and then you let them grow for some amount of time. You will see growth, which is represented by these sort of diagonal lines here on the plate, so bacterial growth, and then you'll see some clearing zones or not clearing zones. And so what are these? So if you see, in this case, this disc A, whatever antibiotic is here, this bacterium that's on the plate when it grows, if it grows right up to the disc, then it doesn't care that antibiotic is there. It is resistant to it. However, there can be varying degrees of sensitivity to antibiotics. So in this case, what we're showing is that there's this clearing zone, also called a zone of inhibition, where bacteria do not grow around that disc. And so, depending on the antibiotic, the size of that zone of inhibition can vary, as it's shown here. For different organisms, that zone of inhibition for the same antibiotic can also vary. This is a really quick qualitative, and it's semi-quantitative depending on how you do it. We're not really equipped to do it in a quantitative way, but you can measure these zones of inhibition to get an idea of how sensitive the organism is to the antibiotic. All right, so now another assay that we're going to do this week is determine the minimum inhibitory concentration of an antibiotic, or the MIC or MIC. You may have heard of this before. And this is another very simple idea where you simply take broth medium and you put antibiotic in at a certain concentration, and then you dilute it. We're going to dilute it, the concentration in half over a number of tubes. And so we'll start out in here. We're starting out with a certain concentration. So here is the concentration. Now, before I go any further, I just want to say that this schematic here is not representative of exactly what we're going to be doing in the lab, but just to give you conceptually an idea of what's going on. Check the lab manual to see how we're actually diluting and what concentrations we're starting with. Okay, so here if we start with 256 milligrams per liter in this sample, it will be diluted in half, through a series. And so we have now the antibiotic in varying amounts in these tubes. Then we have a couple controls. So we have one where we have no antibiotic. So this tube, when we put our bacteria in there, there should be no inhibition of growth. The bacteria should grow because there's no antibiotic. This tells us that the bacteria is able to grow in the media that we're using. Another control is that we put no bacteria into a sample. So that's over here. This is just to show that the medium is not originally contaminated with any bacteria that we don't intend to investigate. Okay, so what do? We make these series of dilution tubes, and we dilute the antibiotic, and we put the same amount of bacteria in each one of these tubes, except of course this first one over on the right, because we don't put any bacteria on there. And then we incubate them overnight. And so we let them we give an opportunity for them to grow. And what we see is we see the tubes lined up here. Wherever we see green in this example indicates that there's been bacterial growth. And what we do is we look for the minimum concentration that inhibited growth. So they were growing, they grew up here to 32 milligrams per liter. And then when there was 64 milligrams per liter, that inhibited the growth. This is the minimum inhibitory concentration, 64 milligrams per liter. Now, if you really wanted to get very specific about what concentration, you can then set up a dilution series between 64 milligrams per liter and 32 milligrams per liter and refine this number. But for what we're going to be doing, we're going to have sort of a very similar mechanism here, where we're going to determine, based on our initial series of dilutions, what that minimum inhibitory concentration is. Okay. Now, a couple things to consider is that the minimum inhibitory concentration for an antibiotic can be different for different bacteria. They don't all, some bacteria have more sort of natural resistance to it and can grow better and withstand higher concentrations. And then also conditions that you grow the bacteria in can affect whether or not how susceptible that bacteria is to an antibiotic. So growing at different temperatures, adding different chemicals, sugars or other kinds of reagents, and see how that affects growth. Now, or inhibition, excuse me. So these are nice sort of assays to use for your projects that you're going to be working on. This is you don't have to do strictly antibiotics. You can do other chemicals that you might be interested in that you think will work as antibiotics. You can do this with the disc diffusion assay too. We have blank discs that you can add different compounds to and see if they inhibit growth. And these are two really nice assays to use for your independent projects to get an idea of what certain compounds might be doing. Okay, now we come to bacteriophage, something that I worked on for a very long time early on in my career. And they're really interesting and fun. So these are simply viruses that infect bacteria. They look sort of like this. They have a head, or called a capsid. There's made up of proteins. Inside this capsid, there is some form of nucleic acid, usually DNA for most of the ones that have been characterized, but they can also have RNA inside them. And then there's some sort of tube, a sheath, and there's different configurations of these. And there's these tail fibers, and sometimes there's these base plates here. And these are the parts that contact the bacteria and say, hey, we found a host of a particular bacterium that's a host that we can infect. And the recognition is going to happen at this end, and the bacteriophage will bind to the cell, as shown here. You can see the capsid and these sort of long tubes interacting with the bacterial cell down here, and they will inject their DNA into the bacterial cell. Now we're going to work with a few different bacteriophage this week, and here's just a couple examples. The one that I showed is very similar to the one that's known as the T-even bacteriophage. T2, T4 is also T6, and a host of other ones, and we have some other ones that we'll be using. There's lambda, which I think is what's on the actual previous picture. Lambda is a very interesting one. It's been used for lots of studies in gene regulation and many other things, as have bacteriophages T3 and T7, which are very, very similar to each other. They're the ones that most look like lunar landing modules. These kill bacteria very rapidly. They have very quick life cycle. And then there's some filamentous bacteriophage. We'll use one of these, M13. There's also one called FD, and there are others that recognize bacteria and infect them. So there's diversity in their morphology and their genetics, the kind of genes that they have inside them, and the size of their genomes. They can be small, from 5 kb up to 500 kb, and I have not checked in a while, but I know that they are new bacteriophage are being found all the time with again more diversity. So here are a few facts about phage. It's estimated that there are 10 to the 31, think about that number for a while, phage in our biosphere. They have a huge influence on the carbon, nitrogen, and phosphorus cycles, and they kill bacteria, which ends up releasing these nutrients. Most of what we know about molecular biology was done using bacteriophage as a model system. Recombination, replication, gene regulation, the fact that DNA is genetic material, was all involved experiments using bacteriophage. Some really interesting things, some of the virulence factors for different bacteria, for cholera and Shiga, there are the virulence factors are actually contained in genomes of bacteriophage that have integrated themselves into the bacterial chromosome. So they are bacteriophage encoded. And bacteriophage have been used for, to treat infections for over, I believe, over 100 years. This has been investigated. And so this term is phage therapy. So bacteriophage kill bacteria. So why not try to use them to stop bacterial infections? And this was used for a very long time in the Soviet Union. They didn't have antibiotics as we did, and so they've been practicing this for a very long time and have lots of different kinds of phage cocktails that treat different kinds of infections. More recently, in the United States and in Europe, companies have been trying to use bacteriophage to treat infections or use them agriculturally, or in the case of intralytics, they have food safety products. They have sprays that consist of virus particles that you can spray on lunch meat to prevent growth of Listeria, E. coli, or Salmonella. And they keep working on getting new kinds of sort of phage cocktails to sort of work on this. Okay, so we're going to talk about two kinds of bacteriophage life cycles, and they can be related. So here we're going to look at our bacteriophage here, coming up and recognizing a bacterial cell, and it's going to inject its chromosome, so the phage genome, into the cell. When it does that, that phage genome is now going to have a bunch of instructions to divert usually the host transcriptional machinery and translational machinery to start away from making the bacterial proteins and transcribing the bacterial RNAs and start transcribing viral RNAs and making viral proteins. And what that does is we end up getting lots of proteins. We get replication of the phage genomes. They get packaged into virus particles, and we get a certain number, and then eventually there's a whole bunch of bacteriophage inside the cell. There are proteins that will poke holes into the cell membrane and cause the membrane to rupture. The phage can then exit the cell, and they go and infect other cells around them. So repeating this process. So there's this cycle. And this is some of the phage that we'll use, T3, T4, and T7. They do this. This is the lytic cycle, and that's all that they do. Now, some bacteriophage, I sort of alluded to this in one of the previous slides, can live a different life cycle. They will infect, so follow this process, and they have their genome in the cell, but now they can do something different. This genome can be integrated or put into the bacterial genome. And when it does, it's called a prophage. So this is the lysogenic cycle. Bacteriophage lambda does this. Bacteriophage P1 can do this. P1 doesn't actually integrate into the genome. It sort of, the bacteriophage genome will sit outside of the bacterial genome in the cell. But it can hang out there for a very long time and be replicated and along with the host chromosomal DNA and be safe there. And then there'll be conditions where it's more favorable for them to go through infection, and they will start to exit the bacterial genome and eventually start entering into this lytic cycle, and they can go through this again. They can go through the lytic cycle as they need to, and then when certain conditions indicate that it's better for them to integrate into chromosome, they can go through this life cycle and stay here for a while, and then eventually go back and forth between them. Okay, so just some notes about what I just said. So the bacteriophage, they need a host. They don't have all the machinery to make proteins. They don't carry ribosomes with them. Some will carry RNA polymerase with them, and a lot of them will carry proteins that will interact with RNA polymerase to alter the transcription profile of an infected cell compared to a non-infected cell. And so for the lytic phage, the virus just takes over the cell, it replicates, breaks open the bacterial cell, and just releases its progeny, and as mentioned, goes and keeps infecting. In lysogenic cycle, the phage DNA is replicated along with the bacterial chromosome. It gets passed on to bacterial daughter cells. And as I mentioned, the DNA can exist autonomously, like a plasmid for phage P1. It can integrate into the bacterial chromosome like phage lambda. There's also a phage in cholera, in Vibrio cholera, that phage CTX5, and that has a couple of the toxins and proteins, has toxin protein and some other virulence factors encoded within it, and is what makes cholera as pathogenic as it is. And just to remind you that this integrated viral DNA is called a prophage, and it can grow and divide stably in the bacterial genome until viral reproduction is induced. So how do we look at bacteriophage? So bacteriophage, so for bacteria, most of you are now used to spreading bacteria onto a plate. You put some bacteria, a culture that has bacteria on it. You either spot plate it or you spread plating, and you spread the bacteria around, and you'll see colonies appear after some time. Well, bacteriophage can't do that. You can't, if you put bacteriophage on a plate, they're not going to grow because they need a bacterial host. And so we have to do something different. What we do is we take and we put bacteria that we want to infect onto the plate first. And we put it so it's evenly spread, hopefully evenly spread across the plate, and then we add our bacteriophage to it so that they can infect those cells that are there. And so here we have a plate with bacteria on it, and we put bacteriophage on there, and the bacteriophage will infect a cell. Eventually, over time, first you'll see some bacterial growth, but then wherever a bacteriophage has landed, it will infect a cell. It will release all its progeny, all its babies will come out, and they will be able to infect the neighboring cells. And you'll see is a clearing zone. It's called a plaque, and that represents wherever a bacteriophage landed and started to reproduce inside cells, make more bacteriophage, infect more cells, do that for a few rounds, and then you get this nice sort of clearing zone. And it looks something like this. You'll see on plates this what's called a lawn of bacteria. So it's an even distribution of bacteria across the plate. So you can't really distinguish any individual colonies. But wherever bacteriophage have landed, you'll see these little holes. Those are the plaques in that lawn of bacteria. And the size of plaques, the way they look, the plaque morphology is something similar to what we've looked at when we looked at colony morphology. Different bacteriophage have different morphologies, and some are very, very striking and characteristic of a particular type of bacteriophage. And these will, depending on which bacterium they infect, they can look a certain way. Depending on the media or the temperature they're growing at, they can have different morphology. So a lot of the same things that we saw with bacterial colonies, there are similar aspects to bacteriophage plaques that they will look different depending on different conditions. So we're going to look at the host range for bacteriophages. So number of different bacteria, the host range is the number of different bacteria that a phage can infect. This is its host range. This is usually limited to a single species for a particular phage. Some have a little bit wider range. Some can only affect very, very restricted strains of the same species. Right? So it's maybe one or two strains, and some are a little bit more promiscuous. And there are a couple of factors that we're going to talk about that limit the host range, like what bacteria a phage can infect. And two of the most important is the phage being able to attach to its host receptors, so things on the outside of the cell recognizing that bacterium as something it can infect. And then I'll talk about one that I like, restriction modification systems that must be overcome. And what I'm not going to talk about, and everybody talks about, is the CRISPR system of adaptive immunity for bacteriophage, and that we may hopefully we get a chance to talk about that at a later time. That's a bit more involved. Okay, so the bacteria have receptors on their surface. These vary in their structure and their location, usually located in the membrane or the bacterial cell wall. These receptors can be altered by a couple different mechanisms. So they can simply be mutated in the genome, and then now they look different, and the bacteriophage can no longer recognize them, and so they can't infect that cell. And also when bacteriophages like lambda lysogenize, when they go integrate into the bacterial chromosome, they could alter the receptors and how the other bacteriophage interact with the surface of the cell. Now, one of the ways that I think is really interesting is this sort of restriction modification system. So many of you have worked with restriction enzymes or have heard of restriction enzymes that are used to cut up DNA for cloning or for different means, and sort of a biotechnology aspect. So these enzymes are naturally occurring, many of them, and they were first discovered as a system that bacteria can use to keep foreign DNA that's not of itself, out of their cells. Because the bacteria has no way of knowing whether a piece of DNA that comes into the cell is good or bad for it. So we're used to using the restriction enzyme, the DNA endonuclease that cuts DNA. But usually in the same operon, there's another gene, which is a methylase, and it's called a modification enzyme. This attaches a methyl group to DNA sequence that is recognized by the restriction enzyme. So they recognize the same exact site, but the modification enzyme puts this methyl group somewhere in the sequence that is recognized. And this alters the interactions between the DNA and the endonuclease, preventing the endonuclease portion of this system from cutting the DNA. So the DNA is now protected. So any DNA that has this modification system is now protected from the endonuclease. And different bacteria have different restriction modification systems, which have been great for biotechnology because many researchers have isolated different ones, and now we have a whole host of restriction enzymes that we can use to cut DNA to our liking to do whatever sort of purpose we need. And so this was worked out. So a lot of this was worked out a long time ago, where there was certain bacteriophage, like T4, that was grown on a particular strain of E. coli called K12. Now, when you took that bacteriophage and you infected another K12 strain, it worked just fine. It infected, it made a whole bunch of babies, and they came out and you got a lot of bacteriophage. However, if you infected a different kind of strain, an E. coli B strain, so it's just a different strain of E. coli, the DNA would come in, it would not be methylated in the proper way, and there are restriction enzymes in that E. coli B strain that would chop up the bacteriophage genome and prevent infection. Now, occasionally, there would be an infection by a bacteriophage grown on K12 that would sort of survive for a while and get modified by this methylation events caused by the restriction modification system. And if that happened, it gets converted from looking like it's from the K strain to looking like it's from the B strain. It will survive and it will proceed with infection. And so the sensitivity at some frequency, at very low frequency, can switch. And where you can start out, the phage is from K12, but it will survive on B, and it'll end up having modifications similar to those that, or identical to those that you get from bacteriophage that infect the B strain. And then these sort of B strain modified bacteriophage can easily infect E. coli B, but will now have a difficult time infecting E. coli K12. So we're going to look at the host range of 8 E. coli B phage. And we're going to use all, well, maybe not all of the gram-negative bacteria in the lab. We expect that the host, we're going to use several of them. We expect that the host range would be limited only to E. coli, but I did an experiment that suggested that for some of these, these bacteriophage, the host range is a bit wider than what's expected. So they can infect some other bacteria that you wouldn't normally think they would infect. And we're going to find out and see if that works again. So the method, I just want to quickly go over this. We'll talk about it more in class and have a demonstration, but we're going to use the double agar method for plating phage. So this comes out to try to solve the problem that you have to get bacteria onto the phage, bacteria onto the plate before you infect with phage. And so we're going to use this thing called top agar. So top agar is similar to the LB agar that we've been using on our plates, except that the percentage of agar is lower than in these normal plates. That's actually, the normal plates are called bottom agar. By lowering the percentage, we can melt this, and we just use a microwave, and then keep it at around 50 degrees and it will stay liquid. What we're going to do is we're going to take some bacterial cells that will already be grown up in liquid culture. We'll add them to the top agar, and when we do this, we usually do one at a time. We take the top agar, add the bacteria to it, and do not place it back into the 50-degree incubator because that can start killing the bacteria and it will heat shock them. And we take the mixture of bacterial cells with the top agar and we pour it onto a plate. As this shows here, we just pour directly onto the plate, swirl the plate around gently to get the top agar to go evenly across the plate, and then we let that sit for a while and solidify. And then once that's solidified, we take, what we're going to do is we're going to take bacteriophage solutions that we already have made up, and we're going to put a small amount in different spots on a plate. I have these cool square plates, and we're going to put the bacteriophage in an order, and then we can see, well, who infects, which bacteriophage can successfully infect whatever bacterial strain that's on here. And what we're going to do is each student will take a different bacterial strain and ask, well, can any of the bacteriophage infect that strain? And do they? And for those that do, is it the same in one strain as compared to another strain? And we'll collect all this data and take a look at it. So we're going to do a lot this week. It's actually not that much work. The biggest amount of work is in doing the minimum inhibitory concentration, because there's lots of serial dilutions. Each student will take one organism and use 2 antibiotics, and your partner will take the same organism and use 2 other antibiotics. And we'll do this for a few different organisms, and we'll check these bacteria against the same antibiotics and see how they react. Then each student will take a particular bacterium, spread it out on a plate, and we have 4 different antibiotics. We have some more if you want to add. We have some other discs. You can add more if you want. And we're going to put discs on that plate. This is really easy to set up. It's one of the beautiful things about the disc diffusion assay. And again, keep in mind that this is something, if you're working with different compounds in your independent project, you may want to use this kind of assay to test them. And then the last thing we're going to do is we're going to do the phage host range. We're going to have one bacterium. We're going to put that in top agar, put it on an agar plate, let it sit for a little while to cool down, add our phage, and then leave it, and then we'll incubate it overnight and look at it the next day.